So unfortunately, our partner in the Eastern Cape, Gwynneth Matcher, who's based at SAIAB, the South African Institute for Aquatic Biodiversity, couldn't make it today to Cape Town. So she sent a few slides, and I'm going to do my best to present them. She said she's available at tea break for WhatsApp questions. So if you have any burning questions, please, you can ask her at the tea break. So yes, she has made these slides. She named it "Teamwork Makes the Dream Work," and yeah, this is a very good example of good teamwork. So SAIAB is one of the NRF institutions. They're all listed here on the slide, and it's based in Makanda in the Eastern Cape. So 3 years ago, Diplomix started a collaboration with SAIAB to enable and to enhance genomics capabilities in the Eastern Cape. So for those of you who've sat in many Diplomix talks before, you'll see that there's a concentration of genomics labs in the Western Cape and in Gauteng. And so we wanted to try and spread that out a little bit more. It's part of Diplomix Spatial Transformation Program. And so we started a 3-year program which is linked to 1KSA. So I'm going to talk a little bit about SAIAB's involvement in 1KSA, and specifically a topotype project, which is almost like a subset of 1KSA under SAIAB, how they're using genome sequencing for eDNA monitoring, and then a bioeconomy project, a tilapia fish farming project as well. So along with all our initiatives, we've also supported a number of different training workshops through SAIAB. Last week, Gwynneth actually ran her Fundamentals in Molecular Biology program. It's the 3rd year that she's offered it, and that program is designed to bring in ecology and biodiversity scientists who don't have a molecular biology background so that they can get up to speed with doing DNA extractions and so on. And as part of our spatial transformation program, we went another step beyond that with SAIAB. So Gwynneth also runs one-on-one training opportunities for staff members and for senior postgrad students from what they call coastal HEIs, coastal historically disadvantaged institutions. So that's Fort Hare, the University of Fort Hare, Walter Sisulu University, and the University of Zululand. So universities that are based along the eastern coast of South Africa. So I'm going to just say a little bit more about that now. So basically this map shows Diplomix supports SAIAB, which is based in the Eastern Cape and Makanda, and in turn SAIAB creates these training opportunities for students and staff members from University of Zululand, Walter Sisulu University, and also the University of Fort Hare. And this is just one of our spatial transformation examples. So the 3rd step of the SAIAB Spatial Transformation Program actually is now in play this year, where we have, in partnership, we have capacitated DNA extraction labs at these 3 different institutions. So in March, we did 2 road trips to these universities to go and meet the students and researchers and the staff members at these labs, and to launch these labs. And literally it's a very basic lab, set of pipettes, qubit, nanodrop, centrifuges, basic DNA extraction facilities. So the plan is that the students that have undergone the training can now undertake DNA extraction in their own home institutes. And we branded this EAGER, enabling access to genomics research, and Science Design and Jacques provided us with another beautiful logo, which we really like, which is shown here on the slide. So watch the space. We really hope that this is going to help 1KSA by bringing in more samples through these different institutions where many of the biodiversity questions are sitting anyway. Okay, so 1KSA. So SAIAB is a major contributor to 1KSA in the marine space. And Gwynneth prepared the slide. So there are over 2,000 fish species in South African waters with 75 endemic fish species. SAIAB researches both marine species and also river species, inland species. So the list over here on the right-hand side are the 19 species that they're currently working with. 12 draft genomes have been assembled, and there are species cards available on the website, including the galjoen, which is down there in the left-hand corner with the South African flag. That's the national fish of South Africa. And so we're very glad to have had this in the 1KSA collection. So a lot of these are still in progress, and SAIAB has plans to expand on these sequencing under 1KSA quite considerably this year, including a number of invertebrate organisms as well. There is a project under FBIP, Foundational Biodiversity Research, called SEA-KEYS, and they are mapping marine invertebrate organisms in the waters around South Africa, and many of these organisms are now going to find their way into 1KSA for genome sequencing. You'll notice on the slide that some of these are critically endangered species, as marked in red, and also endangered and vulnerable. And so it's very important from a conservation perspective that these genomes are created, because it creates tools that can be used in conservation management. So the next project that Gwynneth wanted to mention was using environmental DNA for marine biodiversity exploration. And this slide basically just gives an overview for those of you who aren't familiar with eDNA. Basically, you extract DNA out of water. Water will contain little bits of DNA from cells and tissues and fluids and excretements and so on. And that DNA can then be used through a PCR approach to look for certain tags. And those barcodes or tags can either be generic, so it can pick up all fish. It can be specific for a particular target species, and then you can also obviously target other things such as algae and microbes. So one of the projects that SAIAB is involved in is looking at coelacanths, and I'm sure most of you are familiar with coelacanths. It's a living fossil fish that was found, 70 years ago or so. It's a very interesting story. I'm not going to go into all the details, but just to say that the African coelacanth has been mapped out since that initial discovery, and it's found on the eastern coast of Africa. And it's found in deep caves and canyons, typically between 500 and 800 meters in depth. This is what a coelacanth looks like. It's got these beautiful spots, and as it lives in these deep caves and where it is very camouflaged. In the Isimangaliso Marine Protected Area, which is north of St. Lucia on the KZN coast, the topography of the sea is well understood, and there's Jessa Canyon, which is a deep sea canyon. And SAIAB wanted to understand where are the coelacanths found in this area. It's very difficult to monitor them. It is possible with various craft that can go deep into the water and take photographs, but it is difficult. So eDNA could be a good solution. And what they found is that they could detect the presence of coelacanth using a specific barcode for coelacanth, which they were able to design based on the sequenced genome of coelacanth. So 1KSA hasn't sequenced coelacanth. That was actually sequenced a couple of years ago. It was one of the first South African genomes that was sequenced using the old-fashioned method long before next generation sequencing came along. And what they found is that, yes, the coelacanth is found in Jessa Canyon, big tick over there, and it's found to the south of Jessa Canyon as well. And that fits the Agulhas Current, which flows down that part of the coast. And that's been published as well. Okay, so expanding on their work in Jessa Canyon, they could also use barcoding to look at the biodiversity in that area. And they found all these species of fish, some of which are critically endangered. So it's important to have this knowledge in order to make decisions for management going forward. So the potential impact of eDNA in marine fish research, Gwynneth reminded me that SAIAB has to show the impact always of their research, very much like what Diplomics has to do. So this slide is important. So it can help improve biodiversity assessments for a number of reasons listed there. It can also, it's a non-invasive sampling approach, so it doesn't involve hurting any animals. Once it's set up for a particular species, it is quite cost and time efficient as well, so you can take a water sample quite easily for DNA extraction as opposed to using bigger craft, which are more expensive to run. And it also can be used to detect if there are invasive species coming into an area. So this can be used in fisheries management and also in monitoring marine protected areas. And in particular, there is a coelacanth management plan, and so eDNA monitoring is particularly important for this. Okay, so moving on to the last project now, and this is fish farming. So fish farming is very attractive in South Africa because it can contribute to food security, it could create jobs and more economic growth, and it could also support rural communities. And so these are just some shots showing of what a typical fish farming enterprise would look like. In particular, tilapia has huge potential in South Africa. It is quite an important aquaculture product internationally, but in South Africa it's starting to pick up now. And it's particularly attractive because it's got a high quality white meat content, which is similar to hake. And we all know that the hake stocks in South Africa are under a lot of threat as well. So there are two different types of tilapia which are found in South Africa, the Mozambique tilapia and the Nile tilapia. And Gwynneth made this very nice slide showing the pros and cons of both, the up finger and up thumb and down thumb of both. So the Mozambique tilapia is indigenous to South Africa, whereas it's also adaptable to different environments, and it also has high quality meat, so it would be potentially a good aquaculture source, but it's quite slow growing in comparison to Nile tilapia, which is used more often. So Nile tilapia is used a lot in aquaculture, but it is invasive in South Africa, and it breeds and hybridizes with the Mozambique tilapia, and it can then also outcompete the Mozambican tilapia, and that can cause a problem for river management. So both of these fish have been sequenced already, the genome. And in this project, the plan is it's a part of a bigger project to map the distribution of these two tilapias in Limpopo and in Mpumalanga. And this can be done phenotypically, so by catching fish and identifying which one it is. However, it is quite difficult to tell these two apart. And this is an example of catching the fish. And this is a result of that initial survey as well. So the O. mozambicus, the Mozambique tilapia, is shown in green. The Nile tilapia is shown in yellow. And where there is potential of hybrids is shown in blue, and where there's no fish that they could detect, it's shown in white. So this has all been done on phenotyping. However, genome sequencing could help with this identification. And so the plan going forward is to use low coverage sequencing of these two fish in order to determine which of these scenarios is happening in South African rivers. So are there mixtures of both? Has hybridization occurred, and is that in effect a new species that's developed? Is there partial hybridization or mixed hybridization, or are both cases happening? So this is a very important project. It isn't strictly speaking a 1KSA project, but it uses the techniques that have been developed under 1KSA and a low pass coverage approach to create tools that will help manage this particular problem and opportunity. And just to finish off, Gwynneth would like to acknowledge all the people on this slide who help SAIAB in their research. And I think that's it. Okay, so